Hi friends, welcome to Mother's Teaching. So now I will explain about morphology and structure of chromosome. Introduction to chromosome. Chromosomes are the thread-like structures which are present inside the nucleus of the eukaryotic cells, and it is composed of DNA and proteins. Proteins, those are mainly histone proteins. The main function of chromosome is to carry the genetic information which is passing from one generation to another generation, and the chromosome structure is visible only during the cell division. And the chromosome is discovered by Carl Wilhelm von Nageli in 1842, and the term chromosome was coined by Waldeyer in 1888. So this is the normal chromosome structure. It contains telomeres at the ends in short arm and long arm, and it contains centromere at the center of the chromosome, and also it contains two chromatids. Next, morphology. Morphology refers to the outward appearance. So these are the main five structural features can be seen under the light microscope by staining during the cell division. So those are chromatid, centromere, telomere, secondary constriction, and satellite chromosome. Now chromatid. Chromatid is an arm or half of a duplicated chromosome. Each duplicated chromosome contains two arms, means two chromatids. Here you can see a duplicated chromosome. It contains two chromatids. These two chromatids are very identical to each other, so that's why they are known as sister chromatids. And chromatid is composed of a single linear DNA molecule, and these chromatids are joined at the centromere region, which is present at the center of the chromosome, and these sister chromatids are equally segregated into two daughter cells during the mitosis and meiosis processes, and these are mostly visible during the metaphase of the cell division. Now centromere. Centromere is a region in the chromosome which is located at the center of the chromosome, and it joins the two sister chromatids together, and it is key for chromosomal movement during the anaphase of the cell division, and it is composed of repetitive DNA sequences and proteins. And there is a protein which is formed and present at the centromere that is called kinetochore protein. So kinetochore will forms here. During the cell division, the spindle fibers will bind to the kinetochore of the centromere region, and then sister chromatids will start moving to the opposite poles of the cell. So the centromere helps in the movement of sister chromatids during the cell division. There are two types of centromeres. One is monocentric and holocentric. So in monocentric type of chromosome, it contains only single centromere, and in the holocentric type of chromosome, it contains many centromeres throughout the body of the chromosome. See, this is centriole. The spindle fibers will arise from here. So in holocentric type of chromosome, the spindle fibers will attach to the each centromere of the holocentric type of chromosome. So this helps to flexible and stable separation of sister chromatids during the cell division. But in monocentric type, the spindle fibers will attach it to the single centromere at the center of the monocentric chromosome. So this is little bit unstable separation of sister chromatids when compared to the holocentric type of separation. Next, telomere. Telomere are the regions which are present at the ends of the chromosome, and they are the protective caps of the chromosome. The main function of telomere is to prevent the chromosome from degradation and fusion with other chromosomes, and they are composed of repetitive DNA sequences and proteins. And telomeres shorten with each cell division, and which is linked to aging, means there is a limit called Hayflick limit, which is discovered by Leonard Hayflick scientist, and he told that there is a limit to each cell for the cell division, that is 40 to 60 cell divisions. So for each cell division there is a shortening of telomere ends up to 20 nucleotides. Once the telomeres reach a critically short length, then the cell can no longer divide. So the shortening of telomere which can lead to the cell aging, and this leads to the cell senescence or cell death. So there is an enzyme known as telomerase which can extend the telomeres by adding the repetitive nucleotide sequences to the telomeres to prevent the shortening of telomere during the cell division, and it is associated with the age-related diseases and cancer. Shortening of telomere will link to the age-related diseases and cancer. Now, secondary constriction. Secondary constriction is a region on the chromosome which is visibly constricted or pinched appearance, and it contains NORS, means nucleolus organizer regions, and some genes which are for ribosomal RNA synthesis. And the secondary constriction plays a major role in the ribosome production, and it is only visible during the cell division, especially in the metaphase, and it is found in only some chromosomes but not in the all chromosomes. Now, satellite chromosome. It is a region located near the chromosome and it is appeared as a separated from the main chromosomal body, and it contains the repetitive DNA sequences and including the satellite DNA. Satellite DNA is nothing but the DNA which is present inside the satellite, that is called satellite DNA, and it is mostly found in the short term of the acrocentric chromosome, and the satellite DNA doesn't code for any proteins, and it plays a major role in maintaining the structure and stability of the chromosome. Next, types of chromosomes. Usually there are four types of chromosomes based on the position of the centromere, and those are metacentric chromosome, submetacentric, acrocentric and telocentric. Here you can see in metacentric chromosome it has four equal arms and the position of the centromere is present at the center of the chromosome, and it is appeared as a V shape during the anaphase of the cell division. And in the submetacentric chromosome it has two short arms and two long arms, and the position of the centromere is slightly nearer to one end, and it is appeared as a L shape during the anaphase of the cell division. And next, acrocentric chromosome. In acrocentric chromosome, it has two very short arms and two very long arms, and the position of the centromere is very near to one end, and it has satellite DNA and secondary constriction also. And this acrocentric chromosome is appeared as a J shape during the anaphase of the cell division. And next, telocentric chromosome. Telocentric chromosome doesn't have a short arm, only it contains two long arms, and the position of the centromere is at one end, and this telocentric chromosome will appear as a complete rod shape during the anaphase of the cell division. Next, chromosome size. Basically, chromosome size is based on the total number of base pairs present in the chromosome. And chromosome size is different among the species. For example, human chromosome size ranges from approximately 50 to 250 million base pairs. Some organisms like bacteria can have a lesser number of base pairs, and some organisms like plants can have a higher number of base pairs than compared to the human chromosome size. In human chromosomes, the chromosome number 1 is the largest chromosome and the chromosome number 21st is the smallest chromosome. And chromosome size is measured during the metaphase for accuracy because they are tightly packed and clearly visible. And larger chromosomes contains more number of genes. And size can affect the genetic complexity and function because larger chromosomes can hold more number of genes. This will aiding to the genetic complexity, and this leads to the more functions. Next, chromosome number. These are the main three chromosome numbers. Those are haploid, diploid and polyploid. Haploid means an organism having a single set of chromosomes. For example, gametes like sperm and egg cells in humans having 23 chromosomes. And next, diploid means an organism having a two sets of chromosomes. For example, human somatic cells having a 46 chromosomes total, means they are getting 23 from their each parent, means 23 from their mother, 23 from their father, so total is 46. Next, polyploid means an organism containing more than two sets of chromosomes. For example, in plants like wheat having a six sets of chromosomes and strawberries having a eight sets of chromosomes. And in animals, some amphibians and fish and some other organisms having a polyploid chromosomal number. Now, structure of chromosome. Structure refers to internal organization. Listen, from now this topic is going to be very interesting. So now coming back to the topic, total human genome length is around 2 meters long. As you all know, eukaryotic organisms have a very long DNA, and we are all eukaryotic organisms, right? So human DNA is very long. So that much long DNA have to fit in the very small structure of nucleus. So for that, chromatin is the highly organized structure that can fit within the nucleus of the eukaryotic cell. I will explain you in a simpler way. So this is long linear double helical DNA and this is nucleus. So this much long DNA will not able to fit within the nucleus of the eukaryotic cell. But if this long DNA will be in the condensed form, then this can easily able to fit within the nucleus. So this condensed form is called chromatin. So this much long linear DNA will become into chromatin form to fit within the nucleus of the eukaryotic cell. I hope you understood. And then chromatin is a complex of DNA and proteins. It plays a major role in regulating the gene expression and as well as in the DNA replication. And there are two forms of chromatin. One is euchromatin and another one is heterochromatin. Euchromatin is less condensed and it is transcriptionally active because the genes are present in the euchromatin are loosely packed, so it allows for transcription. So that's why they are more accessible. And while heterochromatin is more condensed and it is transcriptionally inactive because the genes are present in the heterochromatin are tightly packed or densely packed. So that's why they are transcriptionally inactive. Next, organization of chromosome. There are many levels of organization, and organization of chromosome is a highly structured and compact arrangement that allows the long DNA molecules to fit within the cell nucleus while maintaining the accessibility for gene expression, replication and repair also. And then the first level of organization of chromosome is the nucleosome structure, and it is the basic unit of chromatin structure. And nucleosome is the combination of DNA and histone proteins. So in this nucleosome structure, DNA will wrap around the histone octamer. Octa means 8. So this octamer is made up of 8 histone proteins. Those are H2A, H2B, H3 and H4. So two copies of these 4 histone proteins are combined to forming the histone octamer. So this green region is the histone octamer and this red region is the DNA. So this how the DNA will wrap around the histone octamer like this. And the DNA wraps around 1.65 times around the histone core, and the wrapped DNA is stabilized due to the positive and negative charges. Actually, the DNA has a negative charge and the histone proteins have a positive charge. So the wrapped DNA is stabilized due to the attraction between the negatively charged DNA and the positively charged histones. And then H1 protein. So this is the H1 protein. It helps to lock the DNA onto the histone core and to prevent the uncoiling of DNA. This is H3 histone protein and this is H4 histone protein. When the H3 and H4 histone proteins are bounded together forming the H3H4 dimer. Dimer means two, means it has two molecules. Those are H3 histone protein and H4 histone protein. Like this same, this is H2A histone protein and this is H2B histone protein. When these two molecules are bounded together forming the H2A H2B dimer, then the H3H4 dimer along with the another H3H4 dimer are combined together forming the H3H4 tetramer. Tetramer means 4, means it has 4 molecules. Those are 2 H3 histone proteins and 2 H4 histone proteins. So the H3H4 tetramer has a 4 histone proteins. Like this same, the H2A H2B dimer along with the another H2A H2B dimer are combined together forming the H2A H2B tetramer. Then the H3H4 tetramer with the H2A H2B tetramer are combined together forming the H3H4 H2A H2B octamer. Means it has completely 8 histone proteins. Like this, here you can see these are 8 histone proteins are combined together forming the histone octamer, and this is the 147 base pair DNA will wraps around the histone core, and this is the next level of organization after the nucleosome structure. This is the 10 nanometer fiber structure. It is formed by the nucleosomes are connected by the linker DNA. These are nucleosomes, and between the nucleosomes this is the linker DNA. Linker DNA is nothing but the DNA which is present between the two nucleosomes. Those are called linker DNA. So this 10 nanometer fiber structure will appears as a beads on a string like a structure. So the nucleosomes are the beads and the linker DNA is the string. Second level of organization of chromosome is 10 nanometer fiber. I already explained about the 10 nanometer fiber structure, and it provides the initial DNA compaction, and it is the combination of nucleosomes are connected by the linker DNA, and it appears as a beads on a string like structure. The beads are the nucleosomes and the string is the linker DNA. Next, this 10 nanometer fiber structure will be further coiling into 30 nanometer fiber structure, and this is more compact structure than compared to the 10 nanometer fiber structure, and it is formed by the interaction between the H1 proteins of the nucleosomes, and there are two models in 30 nanometer fiber. One is solenoid model and another one is zigzag model. In the solenoid model, the 30 nanometer fiber forms a regular spiral shape and compact the DNA tightly, whereas in the zigzag model, the 30 nanometer fiber forms a zigzag pattern with nucleosomes are stacked in an irregular arrangement. The solenoid model is the regular arrangement of nucleosomes, and in the zigzag model is the irregular arrangement of nucleosomes. Next loop, 30 nanometer fiber will further supercoil into loop-like structure, and these loops are formed when the 30 nanometer fiber is anchored at certain points by certain proteins like cohesin, condensin and SMC proteins. Means the 30 nanometer fiber is holded at certain points by some proteins. Those proteins are cohesin, condensin and SMC proteins. And next, there is a special feature for this loop-like structure, that is one loop contains all the genes which are responsible for one physiological pathway. If that physiological pathway has to be activated, only that loop will be opened, remaining all the structure will be intact. So supercoiling is not just randomly happened. In that supercoiling also there was a engineering in that, leading to the formation of all these structures up to chromosome. And next structure is rosette structure, and loops are further coiled into rosette structure. Rosette structure contains 6 loops, means 6 loops are combined together forming a one rosette structure. It is a higher order structure, and rosette structure are formed by the folding of chromatin loops into a circular or spiral arrangement and organized around a central core. So chromatin loops are folded into a circular or spiral arrangement and organized around a central core. This is how the rosette structure will be formed. Next, coil. Rosettes are further coiling into form a coil structure, and it contains 30 rosettes. Means 30 rosettes are needed for the formation of one coil. And I will show all the images at the last. And next, chromatid. It is a half of a duplicated chromosome connected by the centromere, and it contains 10 coils. One chromatid contains 10 coils, means 10 coils are combined together forming a one chromatid. So duplicated chromosome contains 20 coils because it has two chromatids. For one chromatid, 10 coils are needed. For two chromatids, 20 coils are needed. And next and last organized structure is chromosome. It is most highly condensed structure, and it contains two chromatids, and it is only visible during the cell division. I already explained about the chromatid and chromosome at the beginning of the video. And next, these are the structures of levels of organization of chromosome. Firstly, the double helical DNA. The double helical DNA is further coiled into 10 nanometer fiber structure, that is beads on a string-like structure. And then this 10 nanometer fiber structure is further coiled into 30 nanometer fiber. This 30 nanometer fiber may be either solenoid or zigzag model. Then this 30 nanometer fiber is further coiled into loop-like structure. This is the close-up of loop-like structure. So see how the DNA is arranged inside the loop-like structure, and up to 80 kbp DNA can fit inside the one loop. It is the old picture, so that's why it is mentioned as up to 50 kbp DNA, but up to 80 kbp DNA can fit inside the one loop. And next, this loop is further coiled into rosette structure. I told you one rosette contains 6 loops. So 6 loops, you can count it all also. 1, 2, 3, 4, 5, 6. So 6 loops are combined together forming a one rosette structure. And next, these rosettes are combined together forming a one coil structure. So one coil contains 30 rosettes. So these circles are rosettes. So 30 rosettes are needed for the formation of one coil. And next, these coils are combined together forming a chromatid structure. This is one chromatid and this is another chromatid. So one chromatid contains 10 coils. Here you can see. Um, one chromatid contains 10 coils, but here two chromatids are there. So 2 into 10 coils, total 20 coils. So 20 coils are needed for one chromosome. So I hope you understood. If you like the video, don't forget to like, share and subscribe to Madhu's teaching channel, and stay tuned for more videos, and I will update more soon.